Cool. So thank you so much, everyone, for being here. Thanks to my colleagues that are involved in the BioCat4BioPol project, Daniele Ragno and Tony Giorgino, and a special thank to the panelists that accepted to have a presentation for this webinar. Sebastian Riedel from Berlin. We have Michaela Vannini from Bologna, and we have Gilberto Pereira from Barcelona now. So let's get started. As you know, we will be talking about the project and our experimental activity, and we will have a nice presentation about topics that are related to our project. Especially interesting topic is the AI-assisted enzyme engineering, and also how to face problems in the setting up of the process of production of bioplastics. First of all, I would like to introduce you the group of Verona. The project BioCat4BioPol includes three groups in Italy. One is based at University of Verona, another one at University of Ferrara, and another at CNR in Milano. The University of Verona participates with my group. The group I lead is the biochemistry and industrial biotechnology laboratory. We are based in the Department of Biotechnology of University of Verona, which is a department that has been recognized for 2 times in a row as Department of Excellence by the Italian Ministry of University and Science. So we are currently having extra funds to develop special research line. The first fund was about green chemistry, and now we are more focusing also on agri-food biotechnology. This is the current group. We have some PhD student and postdoc, and also master student that are in their process of running or concluding their master thesis. And of course, all the work done so far is also thanks to the former lab members that now are involved. They are employed in several biotech industries in Italy and also abroad. The people especially involved in the project are Giulia Guglielmi, who recently left. She was hired on the project, and now we have Emma Piccoli, is a PhD student in my group working on the BioCat4BioPol project. Before digging into the topic, I would like to tell you more about what we do in my group. This group is, the main focus is to use industrial facilities, so mankind-made environments as source of robust enzymes. One example is biogas facilities from which we have collected a microbial consortium, and we have selected specific microbial consortium using organic waste from the production of mushrooms. We have selected several microbial communities at different temperature. We work with both microbes and enzymes, both mesophilic and thermophilic, and sometimes also extremophilics. We do analysis of the entire metagenome sequence, and we also test the enzymes that they are expressed by these microorganisms, aiming to identify new enzymatic activities and more robust enzymatic activities, because most of the time the problem is that when you use wild-type enzymes in industrial condition, they fail to work properly. And from this area of research, we have also recently identified a new species, a new microbial strain, a thermophilic strain that we are going to patent. And this strain is able to both hydrolyze lignocellulosic material and ferment it in a one-pot process. Similarly, we have been working on the degradation of plastic. I know that the project is about synthesis, but we want to tackle both synthesis of new polymers and degradation of polymer already out there on the market and unfortunately in our environment. In this project, we used as source of microbes the wastewater treatment plants with the idea that since wastewater is conveyed every day in this system and there are microbes degrading the organic material, since wastewater is contaminated by bioplastics and plastics coming from our clothes and dishes and so on, the microbial community might have developed the capacity to hydrolyze plastics. So again, we enriched several microbial consortia in a mesophilic and thermophilic environment by challenging them with two kinds of polyesters, PET and PLA, as a representative of oil-based, petrol-based, and bio-based polymers. And then we had stable microbial consortia that we have tested for the degradation of polyesters and also for the activity of their enzymes and analytic analysis of their genomes. And from these genomes, the only examples I will talk you about before going to the main topic of the presentation is the discovery of a new PHB, the polymerases, from one of these consortia. This is a model we got with AlphaFold 2. It's a three-domain enzymes with a catalytic domain, a linker domain, and a plastic binding domain. This enzyme has been tested for its activity in gel and in agar plates containing PHP. And as you can see from the cleared zone, the enzymes is active, but in this project that is now being described in a manuscript in preparation, we have also decided to analyze the kinetic of the enzymes on the real substrate, which is something that is quite rare in the literature. Most people, they work on chromogenic substrate. We have done a characterization of the kinetics of these enzymes, both in direct and reverse Michaelis-Menten approach. We understood that the enzymes was better, the kinetic of the enzymes was better described by conventional Michaelis-Menten approach, and we could determine the kinetic parameters on the real substrate. So this is a powder of PHP used as substrate, is not a chromogenic compound. So we have ways, but many groups are working on this, on degradation of polymers, especially on PET and PLA as bioplastics, but we have also to face the production of polymers and the state of the art linked to the project. The idea stemmed from the steep increase throughout the years of the world plastic production. This is a report from the Plastic Fact to 2024. And as you can see, we have a steady increase of the fossil-based production and also a quite more modest increase of mechanical recycled plastics, but you can immediately spot that the circular plastic that include mechanical recycled and bio-based and bio-attribute plastics. As it comes to real bio-based and bio-attribute plastic, we have 0.7% of the total global production, which is an issue. And we have also to recall that bio-based and bio-attribute plastics, they can pollute the environment similarly to what fossil-based plastic do. So besides finding ways to produce this plastic and to replace petrol-based plastics in order to mitigate green gases emission, we also have to set valuable chains to in order not to have the same problem with the bio-based plastics that we had with fossil-based. So first of all, there is an urgent need of bio-based and bioattribute plastics that effectively can degrade into the environment, even though I must say that this is a bit unlikely, because in most of the environment, the temperature does not allow the enzymes to attack the plastics. So some of them, they are compostable, because during the composting process, temperature increase, but a plastic bag, a bio-based plastic bag in the sea will unlikely be easily biodegradable. As it comes to the share of plastic production in Europe, we are doing well. I mean, the global plastic production is 20.3%, but as you can see, the percentage of recycled post-consumer plastic and bio-based and bioattribute plastics increase in the EU, considering 27 countries plus 3. So we are one of the main actors increasing the production of bio-based and also facilitating the mechanical and chemical recycling. But as I said, since bio-based plastic have similar issue in the environment, we must also establish new value chains to and upcycle bio-based bioattribute plastics, unless we don't want to replace plastic islands with bio-based plastic islands in the Pacific Ocean. So getting to the project, the state of the art was that at that time when we wrote the application, and still now, there are two main bio-based plastic of interest. Some other are coming up, but I will talk only about PLA and PHP. Polylactic acid is one of the most used and produced bio-based plastic. Is referred as bio-based because it can be produced by lactic acid produced via fermentation, even though most of the polymerization is still done chemically. The issue with PLA is that it has been shown that it is almost unaltered after 250 days in conventional anaerobic digestion plants. It can degrade better in composting facilities, but it depends mainly on the setup of the process. I've talked with the Italian consortium of composting facilities, and they told me that they are still screening bigger piece of plastic from the organic waste before producing compost. So there is an issue there that consumers are asked to throw away bio-based plastic into the organic waste, but the plastic doesn't really degrade in that conditions. More biodegradable are polydroxyalkanoates, which are produced by microbial fermentation. In this case, the polymerization occurs within the cells, even though several extraction methods require chemicals, but I'm sure that Sebastian will talk more about this. There are less polluting process to extract these polymers. Sometimes consumers refer to one of the most produced polydroxyalkanoates, polydroxybutyrates, as stiff and brittle, so due to its high crystallinity. And when we were about drafting the proposal, the idea was, can we actually mix some of the feature of the PLA with the PHP? There was already something in the literature by saying that copolymer of these two monomers could produce different feature of the polymers, but the challenge there was that no one was able to synthesize alternate copolymers with enzymes. So you can have lactic acid and hydroxybutyrate alternating into the chains by chemical synthesis, but our aim was to have this kind of dimer within the polymers synthesized by enzymes. So this is the graphical abstract of the project. There were and there are promiscuous synthesis enzymes. These enzymes are those that in the cells polymerize polydroxyalkanoates. Some groups have um, engineered these enzymes in order to make them recognize lactic acid instead or together with hydroxyalkanoates. But the issue here is that the polymer that come out, they are still block or copolymer. So there are more elements of the same chemicals in a row. What we would like to achieve is to have an alternate copolymer of this, because what has been reported is that this kind of copolymer, they get degraded at the hydroxyalkanoate area, but the region with PLA are more recalcitrant to degradation. So going to the objective, we wanted to develop a model trained for the discovery engineering on specialized P3HP and PLA synthesis. And soon we will tell you also that we had issue in doing this, because since many people are working with AI, most people are not working in characterizing enzymes, and we need information to feed models. So if very little sequence are characterized out there, you cannot build an AI model, but we found a way to tackle this issue. And of course, one of the objectives was to test the feature of the specialized enzymes. So we first wanted to have specialized enzymes for each monomers, and then somehow try to mix and match the specificity in order to have chimeric enzymes that could alternate the monomers into the polymers. Of course, there is an objective, and Daniele is in charge of this, of the analysis, of course, synthesis of the precursor and analysis and of the composition and the feature of the produced copolymers in order to implement the AI model, to design, as I said, hybrid P3HP PLA polymerases to target the synthesis of alternating copolymer. And then hopefully to scale up the process, at least at lab scale. This is a chart that illustrates the interaction between the different VPs of the project and all the involved entities. As I said, we are two university, one research institute, and also a bio-based consortium working on this project, and each of us is leading a different VP. The VP2 is led by my unit, and the objective is to test catalytic feature of newly discovered select enzymes and provide information to improve the models. The task were to start up activities by the production of biocatalysts and testing assays conditions. So we first starting working on already characterized enzymes in order to set up the proper condition of the enzymatic assays, and then the production of newly identified biocatalysts, and also the production and characterization of the newly discovered AI engineered enzymes. I will just say a few words about our activities before handing the mic to the next speaker. We used a system of a water organic solvent to phase reaction system. In the organic phase, the precursor of the reaction that go to a transesterification reaction with the CoA present in the water phase, so we can have the production of the monomers that are used by the enzymes that polymerize the polymer. So we got these materials from Daniele's group, and we set this condition in order to have the recycling of the CoA at each reaction that could transesterify every time with a different monomer. These are some visual data. This is a negative control, and as you can see, for the 3 enzymes we have tested, we got the polymers that we have also characterized in the composition, and most of the data confirm that for most of these polymer, the main building block is hydroxybutyrate. So even the synthesis reported to be promiscuous failed in the introduction of lactic acid into the chain. The last thing I will show you is a preliminary results. We have tested one of these, especially I think it should be, yes, it's these enzymes as it is, and we have measured the velocity of in this case, we were measuring the turbidity of the reaction medium. And using 10 micromolar of the enzymes, as it was reported in literature, we have found an increase of 0.08 absorbance unit per minute. But then, thanks to Tony Group, we have engineered these enzymes, and just by using 100 times less enzymes, we got just half of the increase of this. So the enzyme is likely to be maybe not 100 faster, but several order. So with this said, I thank you everyone for the attention, and I would like to put your attention to this QR code with which you can also follow the LinkedIn page of the project. And I will hand over the mic to Daniele. I think we can go on and then having the question at the end. Thank you so much.
Yes, Salvatore. Just to let me share my presentation. Okay, can you see it? Yeah?
Yes, thank you.
Okay, so good morning, everyone, and I join Salvatore thanking all of you being here today in this webinar. For us, it's a real pleasure to have an audience like this in this event. Just let me introduce my group. This is the group of the Green Catalysis group of the University of Ferrara. This is a picture taken last summer. Here there is Professor Alessandro Massi, Professor Carmela De Risi, myself, Professor Daniele Ranio, and Professor Graziano Di Carmine. These guys are PhD of the groups, and of course in our labs there are always master thesis and students from different scientific courses. So as you can see from the name of our group, we deal as organic chemists in 2025 in sustainable methodologies in organic synthesis. So we face all the issues related to traditional organic synthesis, and we provide novel solutions in terms of sustainability of process. So our interests are very broad, from photocatalysis to organocatalysis, heterogeneous catalysis, also process intensification, suffix such as flow chemistry. We are very devoted to it. And in this broad area of interest, we always are very interested in materials. So in the application of our methodology in the synthesis of novel material, especially bio-based material, because we were working on valorization of wastes and what's the best way to valorize something, waste, and transform it into a value-added material. So in all this interest, I decided to present you today just our short journey, quite short journey. It was back in 2019 in the polymer chemistry. And that day we were always working with N-tetracyclic carbene. So there were organocatalysts, so metal-free systems that we used for different purposes. At a certain point, we want to add some application to our chemistry. And we say, why? Let's try to use this kind of catalysis in synthesizing micromolecules. And therefore, after reasoning in this way From 2019, we published different work. These are all works from the group, providing new solutions, new strategies in the synthesis of polyesters, polyamides, and also working with dilution conditions, shifting the selectivity towards microcyclic oligoesters that are very valuable intermediate for entropy-driven ring opening polymerization to afford very high molecular weight polymers. And so what's very funny and unique of this methodology was that if we consider the common strategies for step growth polymerization, we can use, of course, a diol, a diamine, working with polyesters, polyamides, but this reactant here is quite not so common because normally you use very activated substrate, diesters, diacyl chlorides, dianhydrides. Well, in this case, we were able to use dialdehydes. And we were able to synthesize a lot of polymers using some bio-based monomers, especially taken from the lignocellulosic matrix, so 5-HMF derivatives. Just few words on the chemistry. I will not be so boring, just to show how it works, because it's something that is shared for all the works. We got this heterocyclic carbene, it's very cheap and easy to generate it with the Popper base. Then this is a carbon-centered Lewis base, can attach an electrophilic dialdehyde, give the Breslow intermediate. This Breslow intermediate can be oxidized with a proper oxidant, very cheap and totally recyclable diquinone, the so-called Karash oxidant. And then you end up with an electrophilic acylazolium that can be intercepted, in this case with the polyester for a diol, and then you end up with your product. And after several catalytic cycles, you observe the chain growth, and you end up with a polymer. This oxidant, the other important thing about sustainability is that it can be used in a stoichiometric fashion and can be totally recovered, or using a very interesting biomimetic system called electron transfer mediator system. You can use iron phthalocyanine with oxygen as terminal oxidant to regenerate the oxidant in your reaction flask. So, as I told before, a lot of polymers were synthesized using also bio-based monomers. So we were using these monomers. Why? Because we knew that, we were aware that novel plastics that were approaching the market, and probably there will also, some of them will also replace some fossil-based one. They were polyfuranoates. Just let's think about PAF that probably will replace PET in the next future. So, as we all know, there are conventional routes like the step growth polymerization using proper monomers, or there was a less common one that was attracting also our attention, that was the synthesis under high dilution condition or pseudo high dilution of microcyclic oligoesters, starting from bio-based monomers, and then ring opening polymerization leading to the high molecular weight products. So, look into this, in this area, we were wondering, it is possible to obtain the direct polymer from the self-condensation of a methylic ester of HMF. You know, everyone has studied HMF in the last like 10 years, I guess. And going back to 1984, there was this example from Moore with very harsh conditions, 240 degrees and antimonium catalyst. It just obtained just black charcoal super cross-linked structure, useless powder. We were wondering if with the mild condition of organocatalysis, we could open a new synthetic window to synthesize this kind of unprecedented polymer. And so we did it. We were able, working with N-heterocyclic carbene with our chemistry under high dilution conditions, to synthesize a mixture of microcyclic oligoesters and then to open them in an entropy-driven ring opening process, organocatalyzed by TBD, obtain for the first time polydroxymethylfuroate. And so with this chemistry, we were able to publish. First publication is 2022, with also in collaboration with Michaela, that will speak later from the University of Bologna. We were able to obtain for the first time this polymer in a fully organocatalytic fashion. And more recent publication, we were able also to introduce a chain extension stage, so increasing the length of the polymer. We were able to, of course, to film it and to study also water contact angle, and always in all these works we always study from all the aspects, the structural point of view, thermal point of view, the behavior of our polymer. In addition, in the last work we also studied an important aspect that was the recyclability. So we tried two different conditions, one of cyclodepolymerization, so ending back with the starting cycles in a circular manner, or otherwise going back to HMFCA, that is another very important building block for polymer chemistry. So now come back to our work, to our pre-war project, the BioCat4BioPol project. This is the team, this is myself, and two PhD students involved in this project, Andrea Odoardo and Francesco Rino. So Salvatore before just introduced what's the role of the unit of Ferrara. We are synthetic chemists. So what we are doing, we are providing the substrate for the tests of the enzyme in the polymerization processes. So we were able to optimize a multi-step synthesis. This is always protecting chemistry, protecting group chemistry that allow us from 3-hydroxybutyric acid and from the methylic ester in both enantiomer configuration of the lactic acid to synthesize the activated monomers, activated as thioesters. So these thioesters are the one that Salvatore, at the University of Verona, uses for the transfer phase reaction. Okay, we optimize all the synthesis on multi-gram scale. We characterize all the substrate, and then there was another very important part of the project that was to convert these thioesters directly in activated coenzyme A esters. So this one was done in purpose, of course, on different scale. It's a small scale, just like 50 milligram scale. Why? To send these compounds directly isolated to UniVOR for water phase enzymatic screening, for more fine screening of the enzyme. So the most problematic part of this work was the isolation. It was done with the collaboration of Professor Salvatore Pacifico of my university. After a little bit of struggle, we were able to isolate with high purity level, confirmed by NMR and analytical HPLC, the products, and also the conversion and the yield of this process was very high, more than 90%. So few waste of coenzyme A that I can tell you is not like the cheapest thing in the world. Another important part of the group is the characterization part. So, of course, when polymers arrive from the test in Verona, we can check from the structural point of view and from the thermal point of view all the features we need to check. So we work a lot with the NMR. We are also very lucky to have a 500 field megahertz NMR that works also in magic angle spinning. Also, maybe if we need to work on solid, we can also do it like that. And we can check monomer composition, of course, also the distribution, if we got an alternating, a block polymer, or a random one. We can also have some access with collaboration to GPC analysis, working, so getting information on site distribution. And of course, we can do classical thermal characterization, so different scanning calorimetry or thermogravimetric analysis to check the thermal stability. Last slide is about an idea that came out working during the project. Nice thing is that we got meeting and we can share new ideas. So let's start from the end of the slide. So our aim was to synthesize an alternating 3HB-LA copolymer, right? So you were wondering, okay, we are always thinking about step growth polymerization, but why not thinking about ring opening polymerization promoted by enzyme? So the idea was to assess this 7-member ring that, of course, got an issue with two possible non-equivalent electrophilic position, but this was something that maybe the enzyme, if you work in a proper way, could manage. But so we started synthesizing this hydroxy acid, and we wanted under high dilution condition to to make a cycle from it, okay, and to end up with this 7-member cycle. But we never obtained this product. While we obtained a mixture of macrocyclic oligoesters with different ring sites, we end up with a yield that was never higher than 30% isolated yield after chromatography, and we characterize it by NMR and AZMS. So we got like dimers, trimers, tetramers, and pentamers. Now we are work in progress trying to push up this yield, and after we hope we can push this wall of this 30% yield, we then work on the ROP, on the ring opening process. So thank you very much. This is enough from my side. I can now give the word to Tony Giorgino.
Yes, good morning, everybody, and thanks to all the attendees. Let me try to share the screen, clicking the right button. Okay, first, a word about my group, which is a computational group working mostly in biophysics. I'm part of the Consiglio Nazionale delle Ricerche, and in particular the Biophysics Institute. But we work in close collaboration with the Department of Biosciences, and in particular with the Master in Quantitative Biology. For example, 2 members of my group, Shams and Julia, come from the said master. There is then Irene Camilla, Federico Ballabio, who is working, actually is the main person working on the BioCat4BioPol project as a postdoc, and then there is myself. In general, our work is at the intersection between biophysics, computation, structural bioinformatics, and statistical inference, which is what powers the idea behind this project. And it's what is most commonly called artificial intelligence, if we want to use that fancy word. The general philosophical approach behind statistical inference is that we follow an inductive process. So just to philosophize, the deductive process is the traditional one, or the most known one from science, which is you start from theory, you formulate hypotheses, and you check the world for observation, and then from there you get or don't get confirmation. So you start from first principles. But the inductive point of view, on the other hand, means that you start from observations. From observation, you form a pattern, and from there you formulate the hypothesis, and from there the theory. The point of view of deduction is that, well, on the right hand you see statistical inference. For deductive reasoning, what's important is that the premises are correct. And if so, the conclusion is certain. For inductive reasoning, it's just probabilistic, which means that we need the data. How do we get to the data? Well, for example, my expertise lies in simulation, in all atom simulations. So we try to model systems, complex systems, in the most reasonably detailed way that can be simulated for times long enough. And it's cursed with the dimensionality problem and its nonlinearity, which means that, as probably you know even better than me, that you deal with the free energy landscapes, which are very rapid. So even if for a given protein or a given process, the most likely state is unique, even if reaching that state from even a slightly different configuration is hard. And that's true both for the simulation, like when we want to make an all atom or a detailed model of the functioning of an enzyme, but also even the process of training models from that. So abstracting their working in the sense, for example, of training artificial neural networks. So this complexity, the ruggedness of the search space is reflected on both sides, both the physics, like I show here, when we try to model a protein as a set of classical forces, including bonds, angles, and dihedrals, but also, on the other hand, when we want to abstract the model as an high-level process. The idea behind most of our works is that we can collect enough simulated data that is realistic enough and covers timescales long enough to reach what I call the thermodynamics scale data sets. So by looking at the trend of the simulation data sets generated for proteins, basically, in the recent decades, it was possible already 10 years ago to extrapolate that by today, we could have reached the timescales of microseconds. So we could sample systems in multiple replicas of the order of 10,000 for lengths that accumulated and reached up to milliseconds, which is very important because it means that we somehow reach the time scale which reflects the actual time scales of biological processes. So if we have sufficient sampling, if our models, our atomistic models are good enough, then it means that somehow we can apply or we can train statistical models, or if you want, artificial neural networks, which could start to represent biological processes like enzyme, conformational changes, folding, and so on, dimerization, and so on, which means that we can also in silico make adjustments to them. So study the effects of mutations and design new proteins. This is, of course, quite time consuming, but the advantage is that if we have large data sets, and here I show some of the applications and the papers published in collaboration with other groups, we can collect, on one hand, data sets which cover systematically many domains of known architectures. And on the other hand, we can build higher level models that simplify them so that we can tweak or make protein design possible. For example, here I show a result from the MDCAT dataset that we published recently. It's a very extensive dataset of 3 terabytes of data in which we systematically simulated 5,000 of the domains classified by the CHT classification. In this way, you can do things like statistically see how the relaxation timescales are affected by the domain class, time, and even temperature. Or in another very large scale study that we did together with the group of Jana Selent, it's family specific for GPCRs. GPCRs are receptors which are target of almost 35% of the current drugs, so quite important. They are extremely versatile, so there are GPCR receptors for any kind of neurotransmitter, lipids, mechanosensors, and so on. And it's important, therefore, to study their conformational transitions, allosteric pathways, and so on. So together with them, we made a very large curated data set covering basically all classes. And from there, inferred timescales of activation, mechanisms, binding sites, and so on. Or another study done with Professor Motta, Monati, and others, we used a classical tool of machine learning, which is self-organizing maps, to build landscapes of the process of binding of a small peptide to its very high specificity receptor, which is the SH2, which is involved in several types of cancer, and we have like 100 copies in our genome, in order to start to both build a map, a geometrical map of the binding process, that you can see on the side, but also a kinetic map that you see on the right. And from the kinetic map, you can also infer not only the bound state that you see in green, but also the transition state. And from there, also the kinetics. Coming closer to the work applied to our PRIN project, we are working with protein design, and nowadays the go-to algorithm for protein design is based on diffusion models. You may have seen for sure diffusion models in use for the process of image generation. So the general idea is that you can take an original data from the distribution of correct data, so to speak, so a real image, if you want, or a real protein, corrupt it in several steps, so adding noise, so moving towards the right, and then at the same time train models to undo your corruption. And that's when you see the time moving towards the left in our animation. So once you train models able to undo the corruption, you can start from basically nothing, plus a conditioning variable, which means, for example, I want to respect a certain region, I want to have a catalytic site, or I want to have an image of a cat rather than a dog, and so on, to go towards more realistic generations. This had a lot of success also in protein design, and this is explained by the intuition had by Ferrutz and others that shows that the basic architecture of proteins has many similarities with the architecture of natural language. So we have an alphabet composed by discrete symbols that assembles into secondary structure, which is words, which it then in turn assembles in higher order structures, so folds, and then it builds meaning by even higher order structures. So the powerful tools of large language models, which have been developed for human languages, have been transferred to the role of proteins, to the world of generative proteins. And in particular, nowadays, one of the most used approaches relies on a combination of tools, which was done by the recently awarded Nobel Prizes. One is called RF diffusion, which generates new structure, condition on the task that you want to obtain. Then you, RF diffusion builds by creating a backbone, an imaginary backbone by orienting several frames of reference one after the other. From there, it is possible to request or to build the side chains, so to infer the sequence which possibly could generate that three-dimensional arrangement. And then other tools are used to check whether that structure is plausible or not, so to screen this huge list of results that you usually get. In particular, now I cannot unfortunately show you more details, but because of our patenting concerns, but Salvatore showed the preliminary results, which were based on the lucky availability of the structure of one PHAC instance from Chromobacterium species USM2, which is available, which was recently crystallized, and which has a complex architecture, and on the basis of which we based our protein design efforts. And this is all from my side.
Thank you so much, Tony. Now I think we can go directly to the presentation of Sebastian Riedel from the Technical University of Berlin. Sebastian, please go ahead. Maybe there is a problem with the connection. Well, I guess we can, okay, cool. We cannot hear you, Sebastian. Okay.
Okay. Yeah, can you see me everything now?
Yes, thanks.
Okay, yeah, sorry for the delay. Yeah. Okay, so short overview over myself. So I started working on PHA bioplastics quite a while ago in Tony Sinski's lab, starting with my diploma thesis 2009, finishing then my PhD 2016, and then built a group at TU Berlin where we have yeah, completed different BMBF projects in this area. And since March 2023, I'm a professor at this Berliner Hochschule für Technik at Berlin. So there we are located, but we are moving this summer in a new building. And yeah, so this is how it looks now. So in summer we should be able to move. So this is quite nice that we yeah, get updated infrastructure. So again, as a short introduction, so we saw the plastic production, the bioplastics production, we only have less than 1% share from the total production from the plastic. We also see that the most plastic is produced in yeah, outside of Europe, increasing every year. And so we focus on PHA. There was still a very small amount, but right now there are a couple of companies, so it's planning to yeah, increase in the next years. So we also saw this. So PHA is like PLA, a plastic which can be produced from renewable resources and is biodegradable. And other than PLA, PHA is completely produced by yeah, many bacteria as internal storage, and is therefore much easier in biodegradation. So in all of these environments, of course, biogas plants, but also even in the ocean, where yeah, cold water and yeah, not so good conditions for further biodegradable bioplastics. So normally it's of course as a linear polyester. So here we see TM of our yeah, strain that we are using, the Solstonia eutropha, and it accumulates PHA when it has unbalanced growth or stress condition and then excess on carbon source. And so there is, you have the yeah, different feedstocks you can use, and then the production of course in the bioreactor, where yeah, we try to get as much as cells with as much as PHA as possible, which is then yeah, extracted from different methods. And then with partners, you also need, like with other plastics, you need additives, you need formulations. So it's like a whole field for itself, and then you need the yeah, construction or the processing into products, and then on the end it should also be biodegraded in the system, and then it can be used again, even as a feedstock. This should be possible. So this is a special material we are working. It's Cuprevidus necator, since this can accumulate up to 90% of PHA as cell derivate. Has a very versatile metabolism, so can use a lot of different carbon sources. You can get very high cell densities, and there are different toolkits available to change the metabolism. And yeah, that's why we are focusing on it. The white type only can produce PHB or PHB co-HB, which has its limitation in its use for several applications. And there we work with some recombinant strain, which can produce also monomers with a longer side chain. And by controlling the monomer amount of the side chains, you can tailor the properties like crystallinity, toughness, melting temperature, and others for examples. So I'm still have access to the lab from TU Berlin, from where my former boss, or colleague now, Peter Neubauer, and there we work on very different topics in this area. The one is we work on biogenic feedstocks, either like waste streams, like from the animal slaughter, animal production or feed production industry, but also from oil production for sugar. So there we can have different systems to screen the different substrates to see how much PHA we can get or in which composition. Also, then we develop tailored processes for bioreactors. So sometimes these feedstocks are very hard to feed to bioreactors, especially at small scale. So that we develop different systems. Also, we work with different PAT solutions, the process analytical technologies like this photon density wave spectroscopy. We come a little bit later to it, which can give us more insights than normal sensors. Then when we have developed the process in a smaller lab scale, we scale it up in our pilot plants, which goes up to 150 liter. And for the data we are showing, we are at 150 gram per liter, where we can produce PHA in a kilo scale for partners to testing. But right now we are also increase this to 300 gram. And this is how the cells look like after drying. We work also on downstream processing, either from dry cells or from wet biomass, and then with partners again for formulation for different kind of products, and also with partners for biodegradation. So also one student of mine, we also work on the strain engineering. So I don't go here in details, but you see to produce the PHA inside the cells, a lot of different enzymes are involved and proteins. And so we were able to identify even new transcriptical regulations. And this knowledge can then be used to get different kinds of PHA or different or new methods to control the metabolism. So this was just recently published, and also she summarized like all the available toolkits for the strains which can be used to make new strains or different products. So one project we had to tailor the content of the copolymer. And so, as I said, our strain needs lipids to make these HHX. We did this over controlling the mixture of sugar and oil. So with sugar, you only get HB. Started like in small scale, and then on the bottom you see that we were able from 0 to here 17 mole%, and this was transferred to a bigger batch reactor even, and then we started also to do like this in fat batch to get this amount of 60 gram per liter, and we're still able to get this controlled composition. And this was scaled up to the pilot plant again, which was successful. But then we increased it in the lab scale to 150 gram per liter, and then went back to the reactor where it was also fits in. So there we can, as I said, produce different in kilogram scale for partners for different applications. So sometimes you need more HHX, sometimes you need less. So it depends also on the application. And this is here the productivity, for example. So then also we did some material characterization with partners, of course. So you see what I said from how the different characteristics from temperature or to elongation to weight, so how long you can stretch it before it breaks has an impact on the HHX concentration, and also some abiotic degradation with the partner. This goes much faster than biodegradation tests, which can give you an idea about the degradation later on. And there we can see, even as I said, PHA is very easy biodegradable. With longer HHX, it gets a little bit slowed down, but it's still, yeah, it still gets degraded. And yeah, so a couple of projects we're working with different animal waste streams. So waste animal fat or products is available everywhere. So here it's in, you see the availability in Europe, and then we work with a company which can separate these materials to hydrolysates of from protein or fat fractions, and then depending on the composition and we use it, or we can get different types of PHA composition. But we were also looking at the fractions which have, for example, a high fraction of free fatty acids, and this is less suitable for other processes like biodiesel production, where you of course can also use fat or plant oils. And also be using the protein fractions, and also we did also some solids. So all different evaluations. Again, this is our what we are interested in, how much HHX polymer we can get, and also an idea if the cells are getting full. So for these fats, they are not liquid, so they are solid. Some have a high melting temperature over 50 degree or more. So we developed a system where we can feed the heat the tubings, and since this is so crystalline, we had to put some lamps that the pumps are not getting clogged, and there we were able then to increase the productivity from these feedstocks from 6% carbon to 17% carbon. And what we also measure is, here you see the cycles which are open. So this is the biomass without PHA, and the fold, you see the total biomass, and when you subtract it, you get you can measure the PHA in gram per liter, and here we always see our HHX content. So then with an interesting feedstocks, this is protein emulgate. This is when you produce the heparin in the pharma industry. You use swine mucosa, and this material contains of fat and oil. And after this extraction, it has like a consist. It looks like the tomato paste when you feed it. So you cannot use it with standard feeding operation. So we developed like a cartridge press, which different Solenoid waves and a computer program, and then we're able to feed this to the bio reactor. Here on top you see the feeding, and which was also interesting was that like over the whole time the cells were full with PHA, and this is due to that the cells can also use the amino acids which are from the protein hydrolysate, and we didn't feed any extra nitrogen in the system, so the cells were triggered always through nitrogen limitation to produce like a high. With every feed bolus, they got carbon and nitrogen, and then they were nitrogen limited all the time. And this maybe we are thinking to use this maybe as a continuous feeding strategy. So then some new work my students put me together is that we are looking to use the waste animal proteins as a replacement for conventional nitrogen sources. But as we see here, so protein hydrolysates are not completely hydrolyzed to amino acids. So we have also longer molecules, and we make an analysis to see the different molecular weights. And what we saw as first is that they might not be able to utilize all of the longer fractions, and therefore we got a little bit less PHA or cell weight from this Schlechenflass experiments. So there's room for optimization, but at least it should be work. Okay. Yeah, so for example here in a little bit in the shadow, you see the reference cultivation with urea as a nitrogen source, and here only with protein hydrolysates. And what we see that at the beginning we have a faster growth with these more rich nitrogen source, but then later it slows up. So maybe this can be used as a booster at the beginning. So, but this is still ongoing. Yeah, what I said about the photon density wave. So when you measure your culture or other samples in optical density, you cannot distinguish between between absorption and scattering. And by applying photon density waves, you can distinguish between that. So you get two signals. You get a signal for absorption, as this is in the blue, and you get a signal for scattering. This is here in red. And what we found out is that the scattering correlates perfectly with our offline cell dry weight. So the cells are scattering and the PHA inside of the cells are scattering, but the PHA is not absorbing. So this we can measure the cell growth without PHA formation with the absorption, and then we get an inline at real time PHA concentration. This is quite nice. So here you see this batch fermentation. You see in the blue we have the growth phase, and we have the PHB fermentation, and the others after 29 hours or something. This process could be stopped. Yeah, but normally you measure PHA over offline with GC, so it takes time. We have no knowledge, and this we transferred also, which is still in preparation, to use the signals to feed carbon and nitrogen to reduce the production time and increase the productivity. Then also we have some online microscopy in the lab where we are able to train the software to see different stages of the cells to link it to different conditions. But it's just I don't have data here in this presentation for that. And for downstream, we have different approaches. So we have focusing on green solvents, like, for example, acetone for the solvent and isopropanol for the precipitant. And after extraction, you separate the cells from the solvent, and you can concentrate the solvent to reduce the consumption. And then you precipitate it, wash it, and then you get the cells. But obviously, this is more expensive than other options, but there you get higher purities. And also there we are working on new systems to get like an inline view on the extraction. So here we compared low field NMR with high field NMR to see the extraction process during the extraction. Here you can see that after, depending, here's the extraction time from 0 to 30. You see that almost after a couple of minutes already, everything is extracted. This can also save some process time. Here may be interesting for you guys who are working so much with enzymes. We are also working on the downstream to simplify the downstream process, because downstream makes up to 50% or more of the PHA production cost. So we have our cell cultivation growth, and then with, for example, high pressure homogenizer or other mechanical systems, we can separate. And there we have different steps we want to include. For example, here some alkalis, or maybe open to use other enzymes to simplify the process to get rid of the usage of solvents. And here in this, all these different conditions we see on the end, it looks that this is almost pure, but when you look at the different, as on the last 2%, or which is not shown here now, that what we also have, or which is the big problem, is the contamination with proteins, nitrogen content of the PHA. So yeah, where to find the right enzymes that the PHA gets purified easier without using solvents is also ongoing interest of our group. Okay, I only put like a sum of my people here. The Björn, he is finished. He works now in industry. Lara and Isabel, they are still here, but they also finished their PhD in December. So Saskia is an ongoing PhD student, and we got 1 or 2 more at the moment. So yeah, at the moment. Yeah. Okay. Thank you for your attention. I hope I was not too fast.
Thank you, Sebastian, for the very nice presentation. I have some questions, but I will wait for the next speaker, the next 2 speaker, and then maybe I can ask more. So I will let the stage to Gilberto. Please, Gilberto, thanks again.
Buongiorno.
Buongiorno.
Hello. So let me just share my screen really fast. Tac. Okay, sorry. And now okay, is everything, do you see it properly? Is it okay?
Yes, we just see a bun on the top, but I think
this here.
Yeah, okay, cool. Thanks.
Okay, thank you. Okay, so thank you for having me today. I'm representing the company I'm currently working at as a postdoc, which is Zymevol. Just a quick introduction about Zymevol. We are a biotechnology company in Barcelona that started off as a dry lab company, and now we have also a wet lab. And our main mission is to use computational methods, including AI, to discover and optimize enzymes for various industrial purposes. In my presentation, what I'm going to try to convince you of is that we have internal technology that we developed and use in both basic research and in projects with clients, where we take a particular enzyme that they're interested in or a particular reaction, and from there on, we do everything up to the discovery of the enzyme, optimization of some properties, and we have quite nice success rate. And finally, I'm going to discuss a little bit about what I'm doing at Zymevol, which is the start of a project on bioplastic, on plastic degradation, specifically polyethylene terephthalate, or PET. Okay, now that the introduction is out of the way, so I think everyone here agrees that enzymes are fundamental biomolecules, and they have many industrial applications. One that I particularly enjoy quite a lot is using enzymes for blood analysis, like for prostate cancer, for example. Another obvious one is lactase, which is very important for people that are lactose intolerant, when you just add lactase to degrade lactose and then make lactose-free milk. But they have applications also as detergents in the medical field, and they can also be used to preserve food. So they're widespread usage, right? The issue is that, well, these enzymes, although they are quite good and they are quite useful, typically they suffer from low catalytic rates. Sometimes they're promiscuous, sometimes they're not very stable, sometimes they're difficult to obtain in large quantities, right? And all of these properties, they are typically the things that our clients come to us and tells us that they need to play with. So they want to improve. For example, thermostability was, up until relatively recently, one of the main issues with PET degrading enzymes. So because when when you have high temperatures, PET becomes amorphous and it becomes more difficult to degrade. But these enzymes, the ones that people were typically using, the PETases, they work at high temperatures. So on one side, you have an enzyme whose optimal activity is at high temperature. On the other side, if you go to that temperature, it's much more difficult to degrade the plastic. So this is sort of the interplay sometimes what we do. Is it possible to improve enzymes? Well, it is. Experimentally, there are two main methods, directed evolution and metagenomics. However, from the literature, we know that this is typically time-consuming because you need to do this over very, very various cycles, and they're costly because you're doing it in the wet lab, right? So it's possible. It has been done before with high success, indeed. But it would be nice if we could, on one side, be more ecological, meaning use less reagents or do less experimental tests, and at the same time be faster, right? At Zymevol, then we took this premise and we thought, well, there's many stages between I have a reaction and I'm interested in, and can I have an enzyme that is efficient for this? And we do this on these different stages using a workflow that we have in-house developed. We start by something we call ZymeScan. ZymeScan is a AI-based method that essentially, based on the smiles of the reagent and the product, it will figure out which enzyme families are likely to catalyze this reaction, right? Then, assuming that we have a good hit, which we normally do, we have BioMatchmaker, and BioMatchmaker is a filtering workflow that takes enzymes from these families and queries them using physical-chemical parameters, using different endpoints that are already in our in-house database. And it boils down to giving you 20, 30 enzymes that may be interesting. Then you filter it a little bit more at the end. So you do a refinement filtering and you end up with a couple of suggestions that may be useful. And then we plug them into Zymevolver. Zymevolver is a method we have that also uses some AI and some machine learning to identify amino acid hotspots for mutations on both at the catalytic site and some other places in the protein surface. And it figures out which mutations are useful. It figures out which hotspots are priority for mutations, so which ones are more likely to lead to an effect that you're looking for, actually. Sorry. And it gives you a ranking of which enzymes are more useful. Normally, we then take this information to our clients and we tell them, well, if you take your enzyme and you do this enzyme, you do these mutations, you're probably going to get what you want. At least we have high confidence in it. And they give us the go-ahead. We have an experimental lab where we do these experiments. And then we use this information to feed a machine learning model to help us go through the next cycles of evolution until we arrive at something that is useful. Typically, this is more or less how it looks like, and Enzyme Evolver is used iteratively between dry lab and wet lab cycles, such that we always have experimental validation of our predictions, and we can back up and we can course correct if we start losing some of the properties that we're interested in. All of this is done in the computer computationally, and so we actually don't test the thousands of enzyme variants we generate. We only test the more interesting ones, which lets us work at a much lower cost than if you do all these tests in the wet lab, and allows us to be much faster because our predictions, they typically take a week or a couple of weeks from start to finish. Just to give you some more details, ZymeScan has been recently updated, and this week we released ZymeScan 2.0. And just for demonstration purposes, I took dichloromethane and I tried to see if I could find an enzyme to catalyze its conversion to formaldehyde. And indeed, we found it. And it's with high score. So ideally here we would take this family and then we'd screen the enzymes that are belonging to this family to figure out if we could optimize, let's say, the catalytic activity. It's very fast. It gives you several enzyme families. Sometimes it only gives you one with high score because there are reactions that are only catalyzed by a very small number of proteins, and all of them within the same family. Well, BioMatchmaker then is our filtering workflow to go from family to enzyme. In the end, after we do the filtering and then we select the most interesting ones, we end up with about 20 enzymes. It takes, let's say, for difficult systems, about 6 weeks. We only had this once, I believe. And then Enzyme Evolver is where we do the mutations and we try to evolve the enzyme we started with towards a given endpoint. And normally we arrive at a list of 90 enzymes because then we can test all of them at the same time in a 96 plate well for example, catalytic activity. And we have already worked with several companies, noteworthy Solvay and Sanofi. And I'm going to show you a showcase of what we did in Sanofi. And our overall success rate is about 80% from start to finish, which I believe that for a computational company that is growing now is quite encouraging. The case study I'm showing you is the regeneration of a PAPS cofactor. For context, so this is a real sulfotransferase 4, AST4, and it typically binds PAP and produces PAPS. So it transfers the sulfate group to PAP. The problem with this is that the PAP to PAPS conversion by this enzyme is very, very inefficient, and PAPS is expensive. PAP is expensive, sorry. And so from an industrial point of view, this is quite suboptimal, right? So you have an expensive reagent and you're not going to produce a lot of the product. However, we figured out that we could use PNPS as a product, as a reagent to give PAP, right? So PNP is much cheaper than PAP, and we can use it to transfer the sulfate group to PAP instead of having an enzyme that catalyzes PAP to PAPS directly. AST4 is this enzyme that is able to do this reaction, so converting PNPS sulfate to PAP to originate PAPS, and that's why Sanofi came to us. Essentially, they had an inefficient process. They found a workaround, but they wanted to make sure that this sulfate group transfer was efficient. So they wanted something that was stable and highly active. We started by doing some mutations on the active site. We considered 16 residues and we did 25 single mutants here. Then we also figured out that there was a channel by which PNPS could enter to the active site. And this channel, we figured out that there were only a couple of hotspots, so we only selected 4 mutants here. We also did an MSA, and from the MSA, we selected some places to do cute mutations. These were called the consensus mutations. They were quite straightforward. And then we also tried to do some distal or allostatic mutations that would induce some allostatic effects on the catalytic site. We ended up with about 90 mutants, exploring 81 hotspots in 1 month for the first cycle. And we found that we tested only 20 of these enzymes, but we found that many of them were more active than the original enzyme. Here, PNP production is being measured because you're removing the sulfate group from PNPS. So you're measuring the production of PNP. And indeed, active site and channel mutations were the ones that actually led to the highest increase in activity. But we thought we could do better, right? Because we could do some dual or triple mutants, and we're interested in seeing if doing this would increase even further the catalytic rate of this enzyme. And so what we did was we selected Sanofi internally selected this variant here that already had 10 distal mutants, and then we did up to 5 more mutations at the level of the catalytic site to generate 90 recombinant proteins. Essentially, we had up to 10, which is the 10, or up to 15, which is the 10 distant mutants, plus this other 5 at the level of the catalytic site. It turns out that we actually improved quite a lot with respect to the original enzyme. So here you have the PNP production rate for AST4 in the wild type. Some of our double mutants here didn't work very nicely, as you can see. Some of them were comparable, but we found one that was much more active. And at this stage, Sanofi was quite happy with the results, and so they decided that this was enough. And eventually, this led to a patent that is here, where over 2 months, we were able to start from this enzyme that was taking PNPS and PAP and converting it to PAPS, leading to a production of an expensive molecule to be used in another pipeline, and with a very low production cost. So this was quite nice. Recently, I was hired in a grant in collaboration with the Spanish government. The idea was that all of this is quite nice, but sometimes our projects fail, and one of the big limitations is that we don't have a lot of dynamic information, structural dynamical information. By this I mean that since it's so expensive to run MD simulations for a large amount of proteins, that is typically impossible for us to reach. And so, however, normally this is done at the all atom resolution. And there are models that allow us to decrease the complexity of these systems by coarse graining. Coarse graining is a method where you are grabbing, for example, the atoms of a chemical group and merging them together into a simulation bead. You reduce the number of particles, hence complexity, so you make simulations faster. while you keep the chemical accuracy. The idea behind this is to make a platform where we can go from docking. Okay, so we try to figure out binding sites for ligands, proteins, binding sites in a protein, for example, where ligands can bind, where polymers can bind, where other proteins can bind. So, and we do this using LightDock, which is AI-based. Is an AI-based method that can do docking of large macromolecular complexes, lipids, small molecules in the current version that is being tested. But we will do it at the coarse grain level, such that we can, for example, try to dock large polymers of plastic in a set of enzymes that are immobilized in a surface, for example. This is one of the ideas of the project. By doing this, we are able to now build a big surface, right? Simulate a this enzyme coated surface with enzymes that are double, triple tetramutants of an original one, and see how plastic polymers interact with each of them. So we can draw dynamical information that allows us to then improve on the design of the enzymes. By building this platform that is a generic platform to study biomolecular systems and also synthetic systems, we think we could extend it to biosensors, enzyme engineering, and drug discovery. So it really becomes a general purpose tool that can be used in combination with the wet lab. My project is basically on these enzyme functionalized surfaces for plastic degradation. The idea is what we were discussing. So there's this monolayer of, let's say it's a silica polymer. We want to immobilize the enzymes to make it easier for degradation to occur. We want to do several mutations, and then we want to study how these mutations affect polymer binding, affect stability of the system, and how can we improve on the properties of the enzyme by drawing also this structural information. We are currently at the level of studying protein-protein and protein-polymer docking because normal docking methods are not equipped to deal with coarse grain models. They're developed for all atom models. This means that we need to, from scratch, optimize all the parameters that docking uses. One of such parameters is the solvent exclusion and the volume exclusion from each protein. And if you do it wrong, you end up with what I have on the right, where one protein goes inside the other, which is obviously not physical. So we're currently working on having this docking method proper and working such that we get good poses to start off. We already tried it, and we used something called the documetrics, which just basically measures how similar you are in terms of your predictions to a crystal structure for benchmarking. And we figured out that for the 8 systems we were testing, we could have good quality solutions already in the first 20 solutions produced. However, this is still not great. It's good from a computational point of view, but from an industrial point of view, you want to have good solutions in the first one or the first 5, because then you looking at 5 structures and figuring out which makes sense and which don't is easy. Looking at 50 is much more complicated, and people will not do it. So we're optimizing this now. Later on, we will also parameterize some polymers. Indeed, in my previous postdoc, I was also already started working with polymers, and I parameterized a strand of PET at the Martini. So Martini 3 is the name of the coarse grain model we are using. And in terms of SASA, in terms of so the solvent accessible surface area distribution, and in terms of the properties of the model I created, I was reproducing quite well the all-atom representation of PET, which here is designed as using the OPLS model for all-atom simulations. This actually was in a paper, so you can search it out. It's called Bartender. Another problem we're facing now is also constructing the surface. Luckily, there are tools that allow us to construct surfaces of any given shape, such as TS2CG or Polyply. And for example, Polyply was used to pack a genome into a capsid, and TS2CG has been used to build nano vesicles, to build curved membranes, to build monolayers, and also to build lipid nanoparticles. So this can be adapted to building silica surfaces to represent, for example, a biosensor. Finally, even if when all of this is done, we still need to decide how we want to immobilize the enzymes. If we're probably going to go by covalent bonds because it's just easier to do. But ideally, we could also optimize the affinity of a part of the enzyme such that it would bind to the silica surface and stay there without an external covalent bond. But we're still studying this. It's a multi-parametric problem, and I've started recently doing taking over this project, so we're still in the very beginning stages. So finally, this is the team. We're about 28. There's some people missing in this photo. I would like to thank all of them. So my main supervisors are Brian here and Ferran. This is Fatima. Fatima is the CEO and also a computational scientist. I would like to extend my thanks to all of them and all of you for your attention. And if you have questions, feel free to ask them.
Thank you so much, Gilberto. Very inspiring and nice data and approach that you have. So I'm sure there will be question later, but now I have to let Michaela to start her presentation.
Okay, thank you so much. I'll just stop sharing. Here. Thank you.
Okay. Let me share my screen. I don't know if it's okay. It's okay. And presentation mode. Did you see my presentation?
Yes, yes.
Okay. First of all, thank you for your kind invitation. I'm a chemist who moved to the field of material science. In my research group, we try to prepare materials in large scale, around 100 grams usually, and we study how it's possible to optimize the polymerization protocols from that should be easily scalable, and how it's possible, yes, to scale up the protocols in order to to transfer our knowledge in the real world, in the real industrial world. So, as already mentioned by Professor Fusco and by Professor Riedel, the plastics represent a global challenge due to their massive volumes involved. Only 10% of plastics are currently recycled, and bio-based plastics account for less than 1%. So to achieve true sustainability, we must push innovation on all fronts, including bio-based plastics. And in this field, Europe plays a key role with with Germany and Italy leading the way, with 75% of all bio-based materials produced in Europe. So this leadership provides a strategic opportunity to further develop the bio-based plastic market. Therefore, to develop bio-based polymers, bio-based plastics, we need naturally derived monomers. I mean, bio-derived molecules having at least two functional groups. In my presentation today, I will focus on furandic carboxylic acid, vanillic acid, ricinoleic acid, and pentadecalactone. Relating to furandic carboxylic acid, since the pioneering studies by Gandini in 1929 and Burgess in 2014, it has been evident that the furan-based polyesters such as PEF, polyethylene furandic carboxylate, and also PPF, polypropylene furan dicarboxylate, are high-performing materials, but their synthesis presents some issues. And for this reason, over the year, research efforts have been devoted to optimizing their synthesis. I report in this table some results related to the PF synthesis, demonstrating that the titanium-based catalysts are really effective in achieving high molecular PEF. But a crucial challenge remains, and this is the formation of DEGF. DEGF means diethylene glycol furan unit, which can be formed by dehydration reaction between alcohol groups during the polymerization. It's easy to recognize the signals due to these groups, this DEG, and the presence of DEG increases the flexibility in the chain. It's very important, it's crucial to evaluate the amount of DEG to understand the final properties of the material. For example, in PET world, the presence of DEG lowers the melting temperature, the glass transition temperature, slows down the crystallization, and makes the polymer less thermally stable. So it's crucial to evaluate, but also to limit the amount of DEGF. We try to prepare PF with low content of DEG, and you can see that a reduction in the catalyst amount, the titanium-based catalyst, and also a reduction in the glycol excess resulted in a PEF with the appropriate molecular weight, but also with the low DEG content. So we have successfully prepared PF, which can be considered the bio-based alternative to PET, mainly due to the similarity in the chemical structure. But is it really the case? Well, Avantium is a Dutch company working on PF since 2010 and received from the European Union more than 22 million euros to build a plant of 5,000 tons capacity of production of furandic carboxylic acid. Why did the European Union invest so much? Because PF, yes, is a real alternative to PET, but not just an alternative, a bio-based alternative. PEF is superior, is characterized by superior performance in terms of mechanical properties, thermal properties and also barrier properties. You see here, for example, PEF is 10 times higher oxygen barrier than PET, is 20 times higher CO2 barrier than PET, and 2 times higher water vapor barrier than PET. And these outstanding barrier properties are due to the lack of symmetry of the furan ring, which prevents the ring flipping along the macromolecular chain. But these incredible superior barrier properties of the furan ring are not only due to the rigidity. You can see here the rigidity is evident also if we consider the glass transition temperature. In this table, I report some data concerning furan-based polyesters compared to PET. Okay, furan is characterized by an inherent rigidity. But another important aspect is the polarity of the furan ring. And this is evident when we consider the water contact angle. In PET, we measured a water contact angle lower than 90 degrees, indicating that PET is a hydrophilic material. This happens because the ester groups present in the structure, in the chemical structure of PET, are able to positively interact with the water molecules. Instead, if we consider PEF or PPF, PF is characterized by shorter repeating unit, PPF longer repeating unit than PET, and therefore if we observe the data for these polymers, in average we have about the same frequency along the chain of ester groups, of polar ester groups, almost the same to PET. Okay, if we consider water contact angle for furan-based polyesters, we measured an angle higher than 90 degrees or equal to 90 degrees. Because with these polymers, the ester groups are not completely available to interact with water. Some of this group are already involved with the other polar interactions, in particular with the furan ring. So the greater number of polar interactions in the furan-based polyesters act as a physical constraints and limit the chain mobility, therefore enhance the barrier properties of the material. But the barrier properties of this material also strongly depend on the crystalline phase. For example, we studied polypropylene furan dicarboxylate, and when PPF is treated at 135 degrees, we observed that develops a crystalline phase, and in particular after 30 minutes, it's visible here with the X-ray diffraction. When the crystallinity increases, we observed that also the barrier increases. In particular, PPF with 35% of crystallinity provides a near total oxygen barrier. And considering the water vapor barrier is reduced to 1/7 respect to the not oriented PET. Therefore, the power of furan ring lies in the rigidity, in the asymmetry, but also in the polarity. These properties together, are very makes the furan very particular molecule, enabling the achievement of these outstanding properties. Another interesting molecule is vanillic acid, which allow us to obtain PEV, polyethylene vanillate. Few years ago, we prepared this polymer and we studied and found that both the PEV amorphous and PET amorphous absorb only a minimal amount of water. And this amount is in any case lower than that absorbed by PET, suggesting better barrier properties of PEV than properties of PET. So PEV can be considered a bio-based alternative to PET. This is for sure due to similarity in chemical structure, but also to similarity in the glass transition temperature, in the melting temperature, not in the crystallinity. PEV is characterized by a very, very high crystallinity, which make the polymer very brittle. Therefore, to exploit its barrier character, the barrier character of the PEV, and limit its brittleness, we tried to introduce along the chain a more flexible monomer. And to do that, we selected the pentadecalactone. So we prepared copolymers according to a protocol developed by our research group, consisting in a simultaneous derivativation of the phenolic, of the less reactive phenolic OH with ethylene carbonate, and then a polymerization by both transesterification and ring opening polymerization. So the whole process is very easy, completely sustainable, because no solvents or purification steps are necessary. Here in this table, I report some data, the chemical characterization of our copolymers. From GPC, the molecular weights are quite high, but in any case, comparable. Through an NMR spectroscopy, it's possible to calculate the molar composition, the length of the unit, and the randomness degree. It's significant that the molar composition, the calculated molar composition, are perfectly aligned with the feed molar amount. Therefore, this indicates the success of our double way of polymerization. Moreover, starting from the monomers, the randomness degree is around 1. So we obtain random copolymers. And we calculated this through an NMR, considering the splitting of the signals produced from the proton color magnet. The red one for belonging to vanillic acid, the violet one belonging to pentadecalactone, and the results were confirmed also by carbon and NMR. Also in this case, we observed the splitting in 4 different signals with different high depending on the molar composition. From the SC, it's clear that only one amorphous phase is present in the copolymers, because only one TG is recorded. And from X-ray diffractions, we deduced that only the crystallization of the prevailing component takes place, and the crystallinity extent decreases in the copolymers. This direct result of this behavior is that the copolymers are less crystalline, so less brittle, and therefore they are processable. I show you in this picture a film prepared made of copolymer containing 80% of vanillic acid. The film is flexible and characterized by good color quality and transparency. Then resinolic acid. This monomer can be derived from castor oil. It contains two functional groups which enable the self-condensation polymerization. A few years ago, we obtained PRA, polyresinolic acid, and we observed that the PRA is characterized by antimicrobial property, intense antimicrobial properties. In particular, we recorded a mortality rate against Staphylococcus aureus after 1 day equal to 100% total activity, total antibacterial activity. The mortality rate was lower against the Escherichia coli and reaches in any case 45%. Unfortunately, PRA is characterized by a honey-like aspect, appearance, and this consistency limits its practical application. So, to solve the issue due to the consistency of PRA, and also to take advantage of its antibacterial properties, we try to combine PRA with PBS. PBS is a biodegradable, bio-based, and semi-crystalline polymer. Therefore, we prepared copolymers through melt copolymerization and physical blends through electrospinning. You can see here that copolymers exhibited only one TG. Instead, in case of physical blends, we found two distinct TG. In any case, all the materials now are in form as powder, so they are processable. And then, considering the antibacterial properties, all the materials result active against the Staphylococcus aureus. The mortality rate is slightly low in case of copolymers for Escherichia coli, but the activity, antimicrobial activity, is very, very high for the blends, probably because PRA is more mobile. And therefore is free to exert its antimicrobial function. The second topic is biocomposites. Biocomposites are considered the perfect solution for a number of environmental issues, such as the depletion of fossil sources, because both the component in the biocomposite filler and polymer usually are derived from natural sources. Plastic pollution, because usually biocomposites are biodegradable, and food waste disposal, because usually, and as filler is used agro-industrial residues. So, as part of two European projects, one called Prolifing, the other one called Know-How, we investigated the effectiveness of natural molecules in imparting new properties to material when added directly to polymeric matrix as filler. So we tested the antioxidant activity of polyphenols extracted from coffee waste. And when we inserted the polyphenols in low amount into PBS, low amount from 1 to 5 weight percent, we obtained antioxidant materials as demonstrated with the ABTS assay. And the antioxidant activity was comparable with that we recorded for the reference, our reference. So PBS containing a commercial antioxidant material, in particular Erganox. We used the Erganox in the same amount, we obtained the same results. Then we used polyphenols extracted from the grape mark waste in blend with PLA to produce food trace. And then, in order to reduce the consumption of polyphenols and also maximize the effect of polyphenols, we tried to apply the polyphenols as coating on PLA films. And we obtain a total antibacterial activity against Salmonella and also Staphylococcus aureus. Even when we applied the coating, the polyphenols as coating on an absorbent pad put in contact with the fresh chicken, we observed a delay in the onset of spoilage by 3 days, demonstrating that the simple incorporation of the polyphenols as filler confers antioxidant functionality to the entire material. In conclusion, we have successfully developed high performance and bio-based polymers and composites, demonstrating the power of natural molecules to impart and improve the material properties. I mean, barrier, antioxidant, antimicrobial properties. These findings represent a step forward in building a more circular plastic economy. And this is all. Thank you for your attention.
Thank you so much, Michaela. Thank you to everyone for the nice insight you have showed us. We are a bit late of schedule, but I'd like to ask the audience if they have question, they can type it into the chat. There is a question now, a general question by Vincenzo Scaiella. He says, in complex research like this kind, what role does a biotechnologist typically play? Which master degree would be the most suitable for working in this field? Does the University of Verona offers a degree program higher than the bachelor that includes courses related to bioplastic production, biofields? What are the bioinformatics courses? But maybe I can take this question later. I mean, I can say a few words. Of course, we have an industrial biotechnology degree and we have a bioinformatics degree. As it goes to the rule of biotechnologies, I think it depends on the choice that you make after your degree. Of course, the degree defines your set of skills, but then I know people that have biotechnology master degree and then they have done computational work or chemical work. So I don't know if some of you wants to add something on this. What would be the best background biotechnologies might have to work in this field? Okay, Vincenzo, if you send me an email, I can tell you more about this, but I would like to ask specific question on the presentation. So while we are waiting for more question to come in the chat, I have a question for Sebastian about the how feasible is to, for example, if we design an enzyme able to perform copolymerization of lactyl-CoA and hydroxybutyryl-CoA, would it be feasible to insert this gene in a strain for the production? Do you think the enzymes would work in the context of a whole cell?
Yeah, okay, good question. I mean, if it's a PHA modified PHA synthase, you mean what can combine?
Yes,
I think
yes.
in general is yeah, it's possible. There are a lot of genetic tools to include it in strains, and then
And then the substrate are there, or you have to also modify the genes to produce the substrates, like lactyl-CoA because these enzymes use CoA activated molecules, right?
yeah, yeah. So I mean, depends on if the strain can naturally use these substrates or not. You know, if
Okay,
it can use it, then you don't need any other enzymes.
okay, okay. And about the measurement of the cell size, so that I don't know, it's a method you have developed, or this is a commercial method, the ones where you check the density of the cells and also the amount of the plastic.
Oh, the PDV spectrometer. Yeah, so we didn't develop the method, so we were working together with another university which develops the system, and a spin-off company from this university. So they are most in the chemical field. And originally, I wanted to measure the emulsion because the oil gets emulsified, and this didn't work. Yeah, and then by accident we find this out.
And
So
so you can get information about the real concentration of the polymer
yeah,
inside the cell
in real time.
in
Yeah,
and it is in line with the data you get later by extracting
yeah,
them.
it's
Okay,
perfectly in line. Yeah,
okay,
so but you can commercially buy this system. But we developed the method. We developed the method how to use it as a control in the bioprocess.
okay.
Yeah.
Well, that's very interesting, especially for the upscale of the process. And then I also have a question for Gilberto about I mean, the tools you were referring to, they are all tools that you can use internally, right? Is there like an open source of some kind?
So, Zyme Evolver and BioMatchmaker, they are internal tools we use.
Mhm.
ZymeScan is open and available for everyone.
Okay.
The idea
Zymescan, okay.
The idea is ZymeScan allows you to boil down the search space of your reaction to a couple of families, right? And then, if you are interested, you can inside ZymeScan, you can contact us directly to ask for example, do you think this is feasible based on my reaction? What would you say the time would be that it would take to do it? Obviously, they're all built using all of the tools we have. They are built using.
We cannot hear you now. You turned off the mic.
Obviously, they are built using physical chemical knowledge, machine learning. We harness multi-sequence alignment data. So, is it possible to build something similar to what we do? Probably. But what we do have that is open, that is our strength is we have used these tools for a long time. We have lots of projects where we apply them. It's a track record of success.
Yeah, sure.
And some of them, for industrial competitiveness, they are locked behind our
but
umbrella. But ZymeScan is free. You can use it. We encourage people to use it.
Yeah, also, it's a way also to get in contact with the company and then
Exactly.
And
actually, some projects actually started like this. So people used the old ZymeScan. They found an interesting result. So an enzyme family they didn't think made sense. And when we dug deeper into it, it actually turned out that the ligand they had was promiscuous.
Okay. So the input you add to ZymeScan is the substrate.
You give the substrate and the product.
Okay.
So as a smile strings. So and then everything is done for you in the web server.
Okay. And you mentioned about some cases that are more challenging than others. So I was curious about to know if the more challenging is the structure itself for the protein or the kind of reaction it performs.
Sometimes the type. So since it's many of this is based on AI, right?
Mm-hmm.
And there's a limited chemical space that one explores normally. If your reaction is something very special, either there's only a couple of enzymes that do it, or we actually don't have an enzyme that does it.
Okay. Okay.
Now, if there's only a couple of enzymes that do it, it facilitates the work because you've already narrowed it down to one or two possible studies. If there's no enzyme to do it, it's more complicated, but also more interesting because it means that you will find something that may work, and if it does, then it's almost automatically patentable, right?
Yeah. Yeah. Cool.
So,
Thank you.
it's a yeah.
But you are okay. I mean, I'm sure I will contact you for European project purposes, so you are open for that, right? In the company to be partner
Yes.
of. Cool. And my next question is to Michaela. I was wondering if you foresee any application of enzymes in the process of synthesis. Do you think?
Not yet.
Not yet. Not yet, but you would be willing to.
Why not? Yes, yes, could be a good solution. But in any case, we must continue with our research line. So we must optimize protocol that can be easily scalable.
Yeah.
So with the enzyme.
It's a bit, it can be a bit tricky sometimes. It depends on the kind of
Now,
process.
probably not in the future, but now it's not so easy.
Yeah, I see. I don't know if Daniele and Tony have questions before we say goodbye.
Michael, I got just one question on the about the polyethylene vanillate. No, so you were telling that the main problem of this polymer is that it's very brittle because it is very crystalline, no?
Yes.
High crystallinity. But as for all the ethylene glycol based polymers such as PF, PET, is not affected by defects from diethyl glycol bridges for the DEG. So, or there is no way to increase the level of DEG to reduce the crystallinity.
When they are present, the crystallinity decreases.
In fact.
So
yeah, I was wondering if there was maybe in the synthetic protocol a way to increase this level of defects, finding a good balance between the crystallinity.
Yes, it's not so easy to find the perfect balance because you have to consider that DAG increase the flexibility and therefore reduce the barrier properties. So these polymers, furan-based and vanillic-based, are usually developed for applications which require the protection. So if you reduce the barrier, is not usually a good choice.
Of course, yeah. So basically, the only route that a chemist can follow in order to increase the performance of this material is the copolymerization.
Yes.
Okay.
Yes. In my opinion, is a good strategy. You have to select appropriate monomer, which can be formed crystalline phase, homogeneous crystalline phase, homogeneous amorphous phase, and that decrease the flexibility, and therefore probably you can obtain a good barrier properties, a good material for the applications you have to develop.
Thank you. Thank you.
Tony, do you have something to add?
No, just a remark that I really appreciated all the presentations and especially Gilberto's. And actually, I wanted to ask him if the light dock that you showed was the project or a software or both?
So, okay, thanks. Thanks for the compliment. LightDock is a tool for docking, for macromolecular docking. And the idea in my project is to insert it into a workflow or a package that allows you to build the surface, dock things into it, and then run the Martini simulations, the coarse grain simulations.
Okay, thank you.
So thank you so much. I don't see more questions in the chat. I would like to take this last chance to thank you all for your time and very nice and productive discussion. I'm sure that more collaboration will stem from this presentation, so we'll for sure keep in touch. And I welcome the audience also to follow the page, the LinkedIn page of the project, so they can be updated about the progress. And I'm sure there will be a final event as well to show the results. So thank you so much. I wish you a nice weekend since it's Friday today, and see you soon. Bye-bye.
Thank you all.
Bye.
Bye.
Thank
you.
Take care.
Bye. Bye.